Good evening and welcome back. Tonight we're exploring something that flows through your veins right now, something carried by roughly 15% of people on Earth, something that perhaps shouldn't exist at all according to what we know about evolution and survival. We're journeying into the quiet mystery of raw negative blood. What is this strange absence that marks some of us and not others? Why does a missing protein create such tangible consequences? Mothers whose bodies treat their own babies as invaders, emergency rooms dependent on rare donors, populations clustered in mountain valleys carrying blood that defies easy explanation. And how did something that causes such dangerous complications during pregnancy manage to persist through tens of thousands of years, surviving bottlenecks and migrations that should have eliminated it entirely? We'll follow the thread from a laboratory in Vienna in the 1930s, where scientists first noticed something peculiar, through the maternity wards where this discovery suddenly explained generations of grief, into the genetic archaeology that tries to trace where this blood came from, and why it clusters so strangely across the world. We'll look at what medicine has learned, what mysteries remain unsolved, and what it means today to carry blood that marks you as part of a small, scattered minority, whose biology refuses to follow the expected rules. As always, I'd love to know where in the world you're listening from, and what time is it for you, whether you're here to drift into sleep or to follow the currents of this biological puzzle. I'm glad you're with me. Now let's begin. The air in the laboratory carried the sharp tang of chemical preservatives mixed with something metallic, that unmistakable scent of blood drawn and stored and studied under glass. Vienna before the war still hummed with scientific ambition. In rooms like these, researchers bent over microscopes in pools of lamplight, hunting for patterns in the invisible architecture of life. The work was meticulous, repetitive, the kind that fills notebook after notebook with observations that mostly confirm what you already suspected, but sometimes in the careful repetition, something appears that shouldn't be there. It's a result that makes you check your work, run the test again, wonder if contamination has crept into your samples. Karl Landsteiner knew this feeling. He'd experienced it before when he discovered the ABO blood group system. The realization that human blood was not all the same, that mixing blood from different people could cause reactions ranging from harmless to instantly lethal. That discovery had won him the Nobel Prize. Now decades later, working across the ocean in New York at the Rockefeller Institute, he was about to stumble onto something equally strange. He and his colleague Alexander Wiener were conducting what seemed like routine immunology experiments. They injected blood from rhesus macaque monkeys into rabbits and guinea pigs, then studied the antibodies the animals produced. Standard procedure. The kind of methodical work that fills scientific careers without drama or revelation. You inject, you wait, you test. Most results are predictable, confirming theories already sketched in textbooks. Except when they tested the antibodies from those rabbits against human blood samples. Something unexpected emerged from the pattern. The antibodies reacted with about 85% of the samples, red blood cells, clumping together in that distinctive agglutination pattern that signals an immune reaction, molecules binding to something they recognize as foreign. But 15% of the samples showed nothing, no reaction, no clumping, as if those blood cells were somehow invisible to the antibodies, as if they were missing something that most humans carried. This shouldn't have been possible according to what they understood about mammalian blood. The antibodies were responding to something on the surface of red blood cells, some protein or marker that the vast majority of humans possess naturally. But a significant minority simply didn't have it. They were missing something fundamental, something so common it appeared to be a default feature of human biology. Yet these individuals had survived, reproduced, passed this absence down through generations without anyone noticing until now. Landsteiner called it the res factor, named after the rhesus monkeys whose blood had helped identify it. If you had the marker, you were Art positive. If you didn't, you were Art negative, a simple classification. A clean binary. But the implications rippled outward in ways nobody fully grasped yet. Because this wasn't like the ABO system where different types balanced each other across populations. This was something else, a trait that most mammals possessed, that appeared universal across primates, yet somehow 15% of humans were born without it. The protein itself sat on the surface of red blood cells like a molecular flag. For most people, that flag was present, marking the cell as self, as belonging. But for those without it, the flag was simply absent. Not damaged, not altered, just gone, as if evolution had reached down and carefully deleted a piece of genetic instruction that every other primate still carried faithfully. Walk through a hospital today, and you can see the blood bank's quiet organization units sorted by type, labeled with letters and symbols that mean everything to the staff and nothing to most visitors. The rant's negative shelf often looks thinner than the others, units parceled out carefully because they're harder to replace. Every pint represents a donor from that 15% minority. Every unit might be the difference between life and death for someone whose immune system will reject anything marked with that missing protein. But in those early days after discovery, before routine testing, before ROGUM, before the infrastructure of careful typing and cross-matching, the implications were just beginning to surface. Doctors had known for years about a mysterious condition affecting newborns, babies born swollen, pale, their bodies filled with fluid, their blood cells destroyed by something invisible. They called it erythroblastosis fetalis. A medical name for a tragedy that repeated generation after generation in certain families. First babies often survived. Second or third babies died more frequently. By the fourth or fifth pregnancy, some women faced almost certain loss. The pattern was clear, but the mechanism was hidden. It was as if pregnancy itself taught mothers' immune systems to attack their own children. But what could possibly drive that kind of response? What evolutionary logic could explain mothers' bodies learning to destroy subsequent offspring? The connection came from a pediatrician named Philip Levine. In the chaos after a stillbirth, he tried to give a grieving mother a blood transfusion to replace what she'd lost during delivery. Routine procedure. The husband's blood matched her ABO type perfectly. But when they mixed the samples, her blood violently rejected his clumping reaction. Incompatibility. Despite having the same ABO group, Levine tested for the newly discovered Ride fact. The woman was Rh negative, her husband was Rh positive. And suddenly the mechanism snapped into focus with terrible clarity. During pregnancy, small amounts of fetal blood sometimes cross into the mother's circulation, especially during delivery when the placenta separates. If the mother is Rh and the baby Rh positive, inheriting the factor from its father, the mother's immune system encounters a protein it's never seen before, a protein it doesn't recognize as self. So the immune system does what millions of years of evolution designed it to do. It mounts a defense. It creates antibodies, specialized proteins designed to attack anything carrying that unfamiliar marker. The first pregnancy usually proceeds safely because the immune response takes time to develop. But once those antibodies exist, they don't disappear. And if the next pregnancy also produces an Rh positive baby, those antibodies cross the placenta and begin attacking the fetus's blood cells, treating them as invaders. The tragedy was written in blood type combinations that looked safe on paper, but created invisible battlefields inside living bodies. An Rh negative mother carrying an Rh positive child. The baby's body working desperately to replace destroyed blood cells. The heart enlarging, trying to pump harder with fewer red blood cells to carry oxygen, fluid accumulating as organs struggle. Many babies were born already dying, their systems overwhelmed by the assault. Before modern medicine, families watched child after child slip away, never understanding why their bodies kept betraying them. Paris records from the 19th century show the pattern: babies born and buried within days, sometimes with notes indicating blood disease or simply failure to thrive. The grief was unfathomable. Try again. Another pregnancy, another death. Some families lost 5, 6, 7 children in succession, each pregnancy becoming an exercise in hope against experience. The evolutionary puzzle cut even deeper. Natural selection is ruthless about eliminating traits that reduce reproductive success. And what reduces reproductive success more effectively than having your immune system kill your own offspring? The Rh negative trait should have been eliminated within a few generations, disappearing like countless other mutations that imposed reproductive costs without compensating advantages. Yet it persisted. Generation after generation, millennium after millennium, Rh negative blood remained part of the human genetic heritage. In some populations it not only survived but flourished, reaching frequencies of 30 or even 35%. This wasn't rare genetic drift. This was something significant enough to withstand the constant pressure of pregnancy complications that killed babies and sometimes mothers. That persistence demanded explanation. Either the trait provided some advantage protection against disease, enhanced immune function, something valuable enough to offset the terrible cost, or it survived for reasons we don't yet understand, preserved in isolated populations where genetic drift could maintain traits that elsewhere would vanish. Either answer suggested there was more to this story than a simple missing protein. The discovery rippled through medicine. Blood banks began testing for Rh factor alongside ABO type. Maternity wards started screening pregnant women. But the questions multiplied faster than answers. Where did this blood type come from? Why did it cluster so strangely across populations? What invisible history was written in the pattern of its distribution? When researchers began systematically mapping Rh-negative frequencies worldwide, they found something that made no evolutionary sense according to standard models of human migration and adaptation. The trait wasn't evenly distributed like you'd expect from a random mutation that occurred deep in human prehistory. It clustered. It formed patterns. Sharp genetic borders appeared between neighboring populations that should have mixed thoroughly over thousands of years of trade and intermarriage. The epicenter sat in a small mountainous region between Spain and France, the Basque country. Among the Basque people, 30 to 35% carry Rh-negative blood. 1 in 3. That's not a rare mutation preserved by chance. That's a defining characteristic of the population, something fundamental to their genetic heritage. To put this in perspective, the global average hovers around 15%. Most of Europe ranges from 15 to 18%. In Asia and Africa, among indigenous populations without recent European admixture, the frequency drops below 1%, sometimes approaching 0. But among the Basques it reaches frequencies found nowhere else on Earth. And the Basques aren't unusual only for their blood. Their language Euskara stands completely isolated from every other tongue spoken in Europe. Not Spanish, not French, not related to any Indo-European language family that linguists have ever successfully classified. It is a linguistic island, preserved through millennia, while waves of Indo-European speakers swept across the continent, replacing nearly every indigenous language in their path. Somehow Euskara survived. Somehow the Basque people maintained their distinct identity through Bronze Age invasions, Roman conquest, medieval kingdoms, modern nation-states. And somehow they preserved Rh-negative blood at frequencies that suggest deep isolation, a population that remained genetically separate while the world changed around them. The correlation was too striking to ignore. Genetic isolation paired with linguistic isolation. A people who somehow stayed distinct while wave after wave of migration washed across Europe. Were they descendants of Europe's first inhabitants, the hunter-gatherers who lived here before agriculture arrived from the Middle East? Were they preserving traits that once covered a much larger territory but survived only in these mountain refuges? The pattern extended beyond the Basques, forming a clear gradient across Western Europe. Move from the Basque country into the rest of Spain and Portugal, and the Rh-negative frequency drops slightly, still high by global standards at 25 to 27%, but lower than among the Basques themselves. Move north into France and you see similar patterns. Western France, especially Brittany and Normandy, shows frequencies around 25%. These are the Celtic regions, the Atlantic fringe, the parts of Europe that remained somewhat isolated from later migrations. Continue north to Ireland and Scotland and the pattern holds. The Irish show Rh-negative frequencies around 25%. The Scots, especially in the Highlands and Islands, similar numbers. Wales roughly the same. All the Celtic regions, all the Atlantic coastal population said, all showing elevated Rh-negative frequencies clustering in the west where mountains and ocean created natural barriers to the great migrations that reshaped the genetic landscape of Eurasia. Then move east and watch the frequency drop steadily, gradually. Germany shows about 15%. Poland similar. Keep going into Russia and it drops to 8 or 10% in some regions. Head south into the Mediterranean and Greece and southern Italy show moderate frequencies around 15 to 18%, but travel further east into Turkey and the Middle East and the frequency drops sharply. By the time you reach India and China, indigenous populations show Rh-negative frequencies below 5%. Africa tells an even more dramatic story. In most of sub-Saharan Africa, Rh-negative blood is extraordinarily rare. In some populations it's effectively absent. Studies of indigenous African populations have found frequencies below 1%, sometimes a fraction of a percent. So rare that when it does appear, Researchers have to consider whether it reflects recent European admixture rather than ancient inheritance. The Americas show the same pattern among indigenous populations with no European ancestry. Pre-Columbian bloodlines carried almost exclusively Rh-positive genes. The Rh-negative blood you find in the Americas today came with Europeans during colonization, mixing into populations that had never known it before. Asia follows suit. Indigenous populations across East Asia, Southeast Asia, and the Pacific Islands show minimal Rh-negative frequencies. Japan less than 1%. China similar. Indigenous Australians virtually zero. Zth is simply absent from most of humanity, appearing at significant frequencies only in one specific corner of the world, Western Europe, particularly the Atlantic fringe. This geographic distribution suggests something profound. The Rh-negative trait isn't ancient in the sense of predating human expansion out of Africa. If it were, you'd expect to find it everywhere humans exist, probably at similar frequencies, with variations due to local selection pressures and genetic drift. But you don't. Evolved and spent hundreds of thousands of years before expanding into other continents. It appears at significant frequencies only in populations descended from a specific region that saw waves of migration, conquest, and replacement over the past 10 to 15,000 years. The conventional explanation invokes migration and mixing. Rh-negative probably arose as a mutation in one population somewhere in Europe or the Middle East, then spread through into breeding. The high frequencies in Western Europe simply reflect the fact that it arose there or arrived early and became established before later migrations diluted it. As Indo-European populations moved west from the Pontic steppe around 5,000 years ago, they mixed with existing European populations. In the east where they originated, their genes are more dominant. In the west they mixed with existing populations that had higher Rh-negative frequencies, creating the gradient we see today. The Atlantic populations, the Basques, Irish, Scots, Welsh, show the highest frequencies because they were least affected by these eastern migrations. They retained more of the ancient European genetic heritage. They're relics, survivors of a genetic pattern that once covered more of Europe before being diluted by incoming populations from the east. This explanation works. It's parsimonious. It doesn't require exotic theories or mysterious origins. But it leaves profound questions. Why would this particular mutation become so common in Western Europe? Blood-type mutations usually remain rare unless they provide some selective advantage. What advantage did Rh-negative blood provide that could drive it to 30% frequency among the Basques? And why doesn't the mutation appear in Africa where humans spent their longest evolutionary tenure? If Rh-negative is just a random deletion of the RHD gene, you'd expect it to appear occasionally in all human populations through independent mutation events. But it doesn't. The clustering is too strong, too specific, suggesting a single origin followed by expansion in one region, while remaining rare or absent elsewhere. Mountains offered protection and isolation. When new populations arrived with agriculture or metallurgy, or conquest, they settled the accessible valleys and plains. The mountain people remained, generation after generation, preserving older genetic and cultural patterns. The Basques in the Pyrenees, the Berbers in the Atlas Mountains, highland populations in Scotland and the Caucasus. These mountain refuges seem to preserve genetic signatures that vanished from surrounding lowlands. If Rh-negative blood is truly ancient, if it represents a marker from populations that inhabited Europe before the great agricultural migrations, then the Basques might be special not because they developed it. They maintained genetic patterns that existed across a much wider territory in deep prehistory. They're a genetic museum displaying traits that elsewhere were diluted or lost through waves of migration and mixing. But this raises another question that science hasn't fully answered. If Rh-negative blood is ancient, if it predates agricultural migrations, why was it advantageous? What environmental pressures during the Ice Age would maintain it at frequencies high enough to reach 30% in some populations? What were our ancestors facing 15 or 20,000 years ago that made this trait valuable enough to persist, despite the pregnancy complications? Europe during the last glacial maximum was harsh. Small populations struggled to survive in landscapes dominated by cold and scarcity. Under those conditions, any genetic variant that provided even modest survival advantages could rise to high frequencies quickly through genetic drift and selection. Small population plus strong pressure equals rapid genetic change. Maybe Rh negative provided advantages we don't fully understand, enhanced immune responses to specific pathogens that are now extinct, metabolic adaptations to cold or to particular diets, something subtle enough to leave no clear archaeological signature but significant enough to tilt the survival odds. Or perhaps the answer is simpler and sadder. Perhaps there was no grand advantage at all. Perhaps Rh negative blood became common in certain populations simply through genetic drift, random fluctuations that happen when populations are small and isolated. Perhaps thousands of families over thousands of years paid the price of pregnancy complications for an accident of genetics that served no purpose, taught no lesson, provided no benefit beyond pure chance. The story changes in 1968. Two teams of scientists working independently, Vincent Frader and John Gorman in New York, William Pollack in Liverpool, developed a treatment they called RAS immunoglobulin. We know it now as ROGAM. The concept was elegantly simple. If you could prevent the mother's immune system from ever recognizing those Rh-positive blood cells during delivery, you could prevent sensitization entirely. No sensitization, no antibodies. No antibodies, no attacks on subsequent babies. ROGAM is essentially a dose of pre-made antibodies against the Rh protein, given to an R. These antibodies quickly bind to any fetal blood cells that entered her circulation during childbirth, coating them and hiding them from her immune system. Before her body can mount its own response, the foreign cells are cleared away. The pre-made antibodies break down naturally. Her immune system never learns to recognize the right protein, never develops that dangerous memory. The impact was immediate and dramatic. Within a decade, deaths from hemolytic disease of the newborn dropped by more than 90% in countries where ROGAM became standard care. Families that would have been torn apart by successive infant deaths could now have as many healthy children as they wanted. The pregnancy paradox that should have eliminated Rh negative blood from human populations had finally been solved through medical intervention. Today the protocol is routine. Every pregnant woman gets her blood type. If she's Rh negative and the father is Rh positive or unknown, she receives Rhyodame around 28 weeks and again after delivery if the baby is confirmed Rh positive. Additional doses follow any event that might cause fetal blood to enter maternal circulation: miscarriage, amniocentesis, abdominal trauma. It's become so standard that most people don't think about it. A simple injection, a few minutes. But that injection represents one of medicine's greatest triumphs, a solution to a problem that had plagued humanity for potentially hundreds of thousands of years, yet the trait remains. The blood type persists. And the questions it raises about human evolution and survival continue to intrigue researchers. Because solved or not, the mystery hasn't disappeared. It's simply moved to a different register, from immediate medical crisis to deeper questions about where we came from and why our blood carries the patterns it does. In hospital blood banks today, staff track Rh negative inventory with special care. They know how fast it's used, how long replacement takes, how catastrophic running dry at the wrong moment would be. Because while most people can receive either positive or negative blood of their ABO type in a pinch, Rh negative patients exist in a narrow lane. For them, especially for women of childbearing age, receiving Rh positive blood risks sensitization, the immune system learning to recognize that foreign protein and building antibodies that could attack future pregnancies. So Rh negative donors carry quiet logistical value. In emergency situations when there's no time for careful typing, O negative blood flows first, the universal donor, the blood with the fewest antigens to trigger immune reactions. Walk into any emergency room during a mass casualty event, and the O negative stock depletes fastest. Those rare donors become crucial infrastructure, living reservoirs maintaining the supply that saves lives when seconds matter. The rarity creates constant tension. Blood donation organizations live with it, sending targeted campaigns to Rh negative donors, urgent requests, phone calls, that subtle sense of being on a permanent list. To be Rh negative in the modern world means carrying hidden value. Your blood not just another data point but a scarce resource, rationed and sometimes desperately needed. Modern research continues probing for patterns. Rh negative individuals show different disease susceptibility. Some studies suggest modest correlations, slightly different risks for certain infections, autoimmune conditions, even mental health patterns. The evidence remains preliminary, contested. difficult to replicate. For every study finding an association, another fails to confirm it when controlling for confounding variables. When COVID-19 swept across the world, researchers turned again to blood type as a variable. Early studies from China and Europe hinted that people with blood type A might face slightly higher risk of severe disease while type O showed modest protection. As for theories, several analyses suggested that being Rh negative might associate with reduced infection risk or milder outcomes. Not immunity, just a gentle statistical tilt. As more data accumulated, the picture grew murkier. Some studies supported the associations. Others found weaker effects or none once age and underlying health were accounted for. The virus was new, the statistics messy. By late 2021, consensus settled on caution. Yes, there might be small correlations between blood type and COVID risk, but they were minor compared to bigger factors like age, obesity, vaccination status. Still, the questions persist. If there are advantages to Rh negative blood, even small ones, they might explain why the trait survived despite pregnancy complications. A fraction of a percent multiplied over millions of people, and multiple generations can shift survival odds just enough to matter. Perhaps in past pandemics, long before modern records, Rh negative individuals fared slightly better or slightly worse. Those stories are buried with our ancestors, written only in the genetic patterns that survive to the present. Meanwhile, another frontier emerges that could transform the entire conversation: synthetic blood. For now, every unit of transfusible blood comes from another human being, screened and typed and stored with strict temperature control and expiration dates. It's a fragile system vulnerable to shortages during disasters, wars, pandemics. For rare blood types, including some Rh negative variants with unusual antigen combinations, the vulnerability is acute. Researchers work on alternatives. Some efforts focus on blood substitute solutions that carry oxygen without using red blood cells at all. Others pursue lab-grown red blood cells, taking stem cells and coaxing them to mature in bioreactors, potentially creating standardized units with controlled antigen profiles. Imagine cells engineered to lack the most immunogenic markers, creating blood that approaches true universal compatibility. Small human trials of lab-grown blood cells have already occurred. The challenges are enormous: cost, scale, safety, regulation. But if synthetic or bioengineered blood becomes routine, it could relieve the chronic pressure on Rh negative donors. No more panic when the O negative shelf empties during emergencies. Yet even in that future, Rh negative blood wouldn't lose significance. To build synthetic systems that work for everyone, scientists need reference points from real donors, including the rare ones. Syria, the 18th word of blood with its stripped-down antigen profile serves as crucial template, helping researchers understand how to minimize immune reactions and design cell surfaces the body reads as safe. The software advantage of being Rh negative in modern medicine isn't a superpower you feel coursing through your veins. It's subtler than that. It's the way your blood fits into the puzzle of human survival at a moment when we finally understand in molecular detail just how fragile that puzzle is. It's the role your rare antigen pattern plays in emergency transfusions, in blood bank logistics, in cutting-edge laboratories where scientists attempt to grow the future in controlled environments. By carrying forward this genetic variation through births and deaths and migrations spanning millennia, Rh negative individuals contribute to medicine's expanding toolkit. Every donation feeds the emergency supply. Every genetic sample adds data points to studies mapping disease risk and immune function. Every pregnancy successfully managed with RhoGAM represents the triumph of knowledge over blind evolutionary forces. So after decades of research and millions of samples analyzed, where does this leave us? What do we actually know about Rh negative blood, and what still sits in shadow just beyond the reach of our current understanding? We know what it is mechanically: a deletion or disruption in the RHD gene that prevents production of the D antigen protein on red blood cell surfaces. We know roughly when it appeared, probably tens of thousands of years ago, not hundreds of thousands, making it relatively recent on evolutionary timescales. We know where it clusters: Western Europe, especially the Atlantic fringe, with frequencies declining as you move east and south. We know it creates pregnancy complications that modern medicine can prevent, but that must have caused immense suffering throughout most of human history. What we don't fully know is why. Why did this trait spread to such high frequencies in specific populations? Why didn't natural selection eliminate something that kills babies? What advantage, if any, offset that terrible cost? The theories range from plausible to speculative. Maybe it provided protection against diseases now extinct. Maybe it was pure genetic drift in small, isolated populations. Maybe it's linked to other traits, immune function, metabolism, neurological characteristics, in ways we haven't detected yet. We also don't know the full range of its effects. Large-scale studies suggest that aside from pregnancy and transfusion issues, rate status doesn't radically alter health outcomes for most people. But Rh-negative is rare enough that detecting subtle effects requires enormous sample sizes carefully controlled for confounding variables. Those studies are difficult and expensive. As we're left with hints, possibilities, patterns that might be real or might dissolve under closer scrutiny, the clustering in isolated populations suggests deep roots in ancient survival. The pregnancy paradox suggests there must be hidden benefits we haven't identified. The persistent fascination it generates across cultures, from Japanese blood type personality theories to Western speculation about ancient bloodlines, suggests it touches something in human psychology beyond mere biology. In the end, I-conclusive blood reminds us that human diversity is wonderfully, irreducibly complex. One small molecular detail, an absent protein on red blood cell surfaces, branches into stories crossing medicine, anthropology, history, and myth. It shapes who can safely receive your blood, how pregnancies must be monitored, what ancient populations you might descend from, but it also reveals our limits. Even with modern tools, genome sequencing, ancient DNA analysis, massive databases, we still can't answer every why. We can trace pathways, map frequencies, model probabilities. Yet evolution's deeper motives often remain half-hidden, like footprints fading into morning mist. The story isn't finished. As more genomes are sequenced worldwide, statistical power to study rare traits like Rh negativity will grow. We may finally detect subtle health effects too small for earlier studies to catch. New techniques in ancient DNA extraction may push further back in time, pulling genetic information from bones that have lain in Earth for tens of thousands of years. We might someday watch Rh-negative variants appear and spread across prehistoric landscapes in reconstructed genetic movies, finally seeing the selective pressures and population movements that shaped its distribution. We may discover it was favored during some epidemic we've never identified, in some ecological niche now vanished. Or we may find something stranger, that it survived through sheer accident, lifted by genetic drift in small populations and maintained through isolation rather than advantage. For now it sits between those possibilities, a real biological anomaly thoroughly mapped yet not fully explained, a trait that complicates pregnancy yet persists, a marker tying together remote villages and modern cities in quiet invisible kinship. One more thread in the vast intricate tapestry of what it means to be human, one more reminder that inside every body beneath skin and story, ancient secrets of our species still flow beat by beat, cell by cell, waiting for future generations to read them more clearly. So now here you are, breathing slowly in the dark, listening to a story written in something you carry every moment, your blood. Whether it's Rh positive or negative, common or rare, it connects you to ancestors who survived plagues and famines and ice ages. Every heartbeat echoes those first small bands around fires under unfamiliar stars. Every drop carries memories of migrations and survival and genetic gambles that paid off in the quiet miracle of you simply being alive tonight. Some mysteries live in distant galaxies. But some of the deepest ones are much closer inside your veins, in the way one tiny protein can decide whether a pregnancy is easy or complicated, in the way modern medicine learned to make peace between a mother's immune system and her child's blood. Your body is full of unsolved puzzles, small everyday miracles scientists still work to understand. You are quite literally a walking archive of evolutionary experiments reaching back hundreds of thousands of years. If you feel curious, you could look up your blood type if you don't know it, ask your parents or grandparents, trace the patterns, see how something invisible connects relatives who lived and died long before you were born. Then you don't have to remember every detail or solve any mystery. You can just let it settle, let the images fade. Ancient laboratories, hospital rooms where tiny vials of medicine, maps showing strange genetic borders across landscapes where blood types cluster and thin. Feel your own heartbeat, steady and patient, older than any legend, the blood that puzzled scientists, that shaped royal lineages, that defied extinction. It's all the same river, and tonight it carries you gently towards sleep. Rest well. May your thoughts be soft and your body remember in its own quiet language that you are part of a lineage of survivors stretching back through deep time. Good night. But you're a woman standing in a doctor's office sometime in the 1950s, holding a small card with letters and symbols that might as well be hieroglyphics. The nurse has just explained that she's Rh negative, that her husband is Rh positive, that there could be complications. But the words slide past without purchase because nobody has explained what those complications actually mean, how they happen, what invisible war might unfold inside her body during a pregnancy. She goes home. She tells her mother, and her mother's face changes. A shadow passes across it, a memory of something not spoken about. 3 babies, her mother says quietly. 3 babies between you and your older sister. All of them born too soon or too sick, all of them buried before they could be named. The doctors called it bad blood back then. They said some women just couldn't carry. They said it was God's will or weak constitution or nerves. But now there's a name for it. A reason written in proteins too small to see. This was the reality for countless families before the mechanisms were understood, before prevention became possible. The grief was enormous and it was silent. Women blamed themselves. Families whispered about curses or punishment. Doctors offered sympathy but no explanation. And the blood type that should have vanished under such brutal reproductive pressure somehow persisted, generation after generation, carrying with it both mystery and sorrow. The mechanics are cruelly simple once you understand them. During pregnancy, mother and baby maintain separate circulatory systems. The placenta acts as interface, allowing oxygen and nutrients to pass from maternal blood to fetal blood without the two ever actually mixing. It's an elegant solution to a complex problem, a biological barrier that protects both mother and child from immune reactions that could destroy either one. But biology is messy. Perfect barriers don't exist in living systems. In small amounts of fetal blood sometimes cross into maternal circulation, especially during events that disrupt the placenta's integrity. A fall, an accident, medical procedures like amniocentesis that pierce the amniotic sac, and almost always during delivery itself, when the placenta separates from the uterine wall and maternal and fetal blood briefly commingle in ways they never did during the months of pregnancy. For most women, this mixing causes no problems. If mother and baby share the same Rh status, the immune system recognizes the fetal cells as self, or close enough to self that no alarm bells ring. But if the mother is Rh negative and the baby Rh positive, those fetal cells carry a protein the mother's immune system has never encountered before, a foreign antigen. The immune system evolved over hundreds of millions of years to detect precisely these kinds of molecular differences. It's exquisitely sensitive to foreign proteins, capable of distinguishing self from non-self with extraordinary precision. This sensitivity keeps us alive, protecting against bacteria and viruses and parasites that would otherwise overwhelm our bodies. But pregnancy asks the immune system to do something paradoxical to tolerate a genetically distinct individual growing inside the body, sharing blood supply and nutrients, for 9 months. Most of the time the system manages this through elegant regulatory mechanisms. The placenta secretes factors that suppress local immune responses. The fetus develops in a partially immune-privileged space. But when Rh-negative mother meets Rh-positive fetal blood, those protections sometimes fail. The immune system sees the D antigen, recognizes it as foreign, and begins the careful orchestrated process of mounting a response. First come the Scout's immune cells that sample the foreign antigen, process it, and present it to other immune cells that can generate antibodies. Then comes the production phase. B cells begin manufacturing proteins specifically shaped to bind to that Rh antigen. These antibodies circulate through the bloodstream, searching for their target. During the first pregnancy, this process takes time, weeks or months. By the time antibody levels rise significantly, the baby is usually already born, safe, unaffected, but the immune system has memory. That's one of its great evolutionary advantages. Once it learns to recognize a pathogen or foreign protein, it remembers. B cells that produced anti-Rh antibodies don't all die off after the first pregnancy. Some become memory cells, long-lived sentinels waiting in lymph nodes and bone marrow. If that same antigen ever appears again, they're ready to respond much faster and much more strongly, so the second pregnancy begins. Another Rh-positive baby, inheriting the father's Rh-positive gene just like its older sibling, and this time the mother's immune system is primed. Those memory cells detect even tiny amounts of fetal blood crossing the placenta. Within days or weeks, antibody production ramps up. These antibodies are small enough to cross the placenta themselves, small enough to enter fetal circulation. Once in the baby's bloodstream, they do exactly what they're designed to do. They bind to red blood cells carrying the right antigen and mark them for destruction. Other immune cells recognize these antibody-coated cells as targets and begin destroying them. The baby's red blood cell count drops. Anemia develops. The baby's heart works harder trying to compensate, pumping faster to deliver oxygen with fewer red blood cells available. As red blood cells break down, they release hemoglobin, which gets processed into bilirubin. Normally, the liver clears bilirubin from the bloodstream, but a fetus's liver is immature, and when hemolysis happens too fast, the system becomes overwhelmed. Bilirubin accumulates. It crosses into the brain where it's directly toxic to developing neurons. This is kernicterus, a form of permanent brain damage that before modern medicine left countless children with cerebral palsy, deafness, intellectual disability. In severe cases, the baby's entire body swells with fluid as the cardiovascular system struggles. This is hydrops fetalis, the most extreme manifestation of hemolytic. In Syria, the babies born in this condition died within hours despite doctors' best efforts. Others survived but faced lifelong disabilities from the period of oxygen deprivation and toxic bilirubin exposure during critical developmental windows. The cruelty compounds with each pregnancy. Third babies face higher risk than second. Fourth pregnancies become terrifying. Some women's antibody levels rise so high that babies die in utero, miscarried in the second or third trimester. Others are born so profoundly anemic they cannot survive outside the womb. The pattern becomes predictable, expected. Doctors in the early 20th century would counsel Rh-negative women married to Rh-positive men about the odds, about what to expect, but they had nothing to offer except sympathy and the suggestion that perhaps limiting family size was wise. Think about that reality. Evolution spent millions of years optimizing human reproduction, developing systems to protect pregnancy and maximize infant survival. Yet here was a blood type combination that actively worked against successful childbirth, that made second and subsequent pregnancies progressively more dangerous. By every rule of natural selection, this should have been eliminated quickly. Any trait that reduces reproductive success faces strong negative selection pressure. But Rh-negative blood didn't disappear. In some populations it thrived. This paradox drove researchers to search for hidden advantages, benefits that might offset the reproductive costs. The search continues today, complicated by the fact that we're looking for advantages that might have existed in past environments very different from modern ones. A trait that's neutral or slightly harmful today might have been protective 10,000 years ago against threats that no longer exist. Some researchers have focused on infection. Blood type affects susceptibility to certain pathogens in ways we're only beginning to understand. The ABO blood group influences risk for cholera, plague, malaria. Perhaps the Rh system does something similar. Early studies suggested Rh-negative individuals might show different patterns of response to Toxoplasma gondii, a parasitic infection common in environments with cats and undercooked meat. Toxoplasma is interesting because it affects behavior in subtle ways. Infected rodents lose their fear of cats, making them easier prey and allowing the parasite to complete its life cycle. In humans, the behavioral effects are more controversial, but some research suggests infection might influence reaction times, risk-taking behavior, even personality traits. If Rh-negative individuals respond differently to Toxoplasma, either more resistant to infection or less affected by its behavioral influences, that could represent a selective advantage in certain environments. Other work has examined viral susceptibility. Do Rh-negative individuals fight off certain viruses more effectively? The data remains sparse and contradictory. Some studies hint at patterns, others fail to replicate them. Part of the problem is that Rh-negative is relatively rare, so studies need very large sample sizes to detect modest effects. Many existing health databases weren't designed with blood type as a primary variable, so researchers often work with incomplete information, Autoimmune diseases show interesting patterns. Some conditions appear slightly more or less common in Rh-negative individuals depending on the specific disease. But teasing apart cause and correlation is fiendishly difficult. Is Rh status directly influencing immune function? Or do Rh-negative individuals share other genetic variants that happen to cluster together because of population history? The answer likely varies depending on which population you're studying and which disease you're examining. Then there's the possibility that any advantage was temporary, tied to specific historical circumstances that no longer apply. During Ice Ages, European populations faced extreme environmental stress. Small groups survived in refugee or ice-free pockets, where game and plant food remained accessible. Under those conditions, even small advantages in disease resistance, cold tolerance, or metabolic efficiency could make the difference between survival and extinction. Perhaps Rh-negative provided some benefit during those bottleneck periods. Perhaps it rose to high frequency through genetic drift, random fluctuations in small populations where chance rather than selection determines which genes become common. Once established at high frequency, the trait could persist even after environmental conditions changed, maintained by population structure and isolation, even though it no longer provided active benefits. The mountain populations tell part of this story. The Basques in the Pyrenees maintaining language and genetics through millennia of surrounding change. Celtic populations in Ireland, Scotland, Wales, preserving high Rh-negative frequencies in regions where geography limited mixing. Berber populations in North African mountains, showing elevated frequencies distinct from surrounding groups. Mountains create barriers. They isolate. They allow old patterns to survive long after they've been replaced elsewhere. But isolation alone doesn't explain why Rh-negative reached such high frequencies in these refugia. Genetic drift can preserve rare traits, but it can also eliminate them. For Rh-negative to hit 30% in the Basque Country requires either very strong drift in a very small population, maintained over very long time, or some selective pressure, however slight, that favored its increase. The pregnancy complications should have provided strong negative selection, pushing the frequency down. For it to rise despite that cost, something else must have been pushing it up. Either that something was selection-direct advantage in survival or reproduction, or it was drift in populations so small that random sampling effects overwhelmed selection pressures. Or some combination, with drift doing most of the work in small isolated groups while modest selection prevented the frequency from drifting back down. We may never know with certainty. The past doesn't leave detailed records of its selection pressures. Ancient DNA can tell us what frequencies existed at different times, but it can't directly reveal why those frequencies changed. We're left inferring from patterns, building models, testing predictions. Science at the edges of knowledge always involves this kind of detective work, assembling fragmentary clues into narratives that fit the evidence without claiming more certainty than the data supports. What we can say is that the pregnancy paradox was real and devastating. Medical records from before Rogam document the toll. In some hospitals during the 1940s and 50s, hemolytic disease of the newborn accounted for thousands of infant deaths annually. Obstetric wards developed exchange transfusion techniques where they would replace a baby's entire blood volume, literally draining out the antibody-laden blood and replacing it with Rh-negative donor blood that wouldn't be attacked. This procedure was dangerous, technically demanding, traumatic. It required multiple IV lines in a newborn's tiny veins. It took hours. Many babies didn't survive the procedure itself. But for those who did, it offered the first real hope of survival. Before exchange transfusion, babies with severe hemolytic disease simply died. There was nothing medicine could do except watch and try to keep the family comfortable with their grief. The development of Rogam changed everything, not just for individual families but for our understanding of what medical science could achieve. Here was a problem that had existed for millennia, causing immeasurable suffering, and within a few decades of identifying its cause, medicine had developed effective prevention. The story moves from mystery to mechanism to treatment with almost cinematic efficiency, but that progression only happened because researchers were willing to look at the problem from multiple angles. Landsteiner discovered the Rh factor through basic immunology, studying antibody responses in rabbits with no particular medical application in mind. Levine connected it to pregnancy complications through careful clinical observation and pattern recognition. Freda, Gorman, and Pollack developed ROGAM by thinking about how to interrupt the sensitization process at its source. Each step required different skills, different perspectives, different kinds of thinking, pure research, clinical observation, translational medicine. The solution emerged from collaboration across disciplines, from researchers willing to share findings and build on each other's work. It's a model of how science works best, how problems that seem intractable can yield to sustained intelligent effort from multiple directions. Today the protocol seems almost mundane in its simplicity. Blood type card filled out at first prenatal visit. If Artra negative, schedule RNGame injection around 28 weeks. Repeat after delivery if baby tests Artra positive. Additional doses after any event that might cause fetal-maternal hemorrhage. Simple guidelines, clear protocols, dramatic results. The kind of public health intervention that prevents so much suffering. It becomes invisible. We no longer see the thousands of babies who would have died but now survive healthy, yet in parts of the world without access to modern prenatal care. The pregnancy paradox continues. Hemolytic disease still kills babies in regions where ROGAM is unavailable or unaffordable. The medical solution exists but distribution is uneven. This is true of so much modern medicine. We have treatments that work remarkably well, but getting them to everyone who needs them remains an ongoing challenge involving economics, infrastructure, education, political will. For women in wealthy countries with good healthcare access, being Rh negative no longer means fearing pregnancy. It means a few extra appointments, a couple of injections, slightly more careful monitoring. The risk has been reduced to something manageable, no more concerning than dozens of other factors. This represents an enormous victory, the kind that's easy to take for granted because it happened before most people currently having babies were born. But talk to someone who lost babies before ROGAM became available and you hear a different story. The fear was real. The not knowing why this kept happening, the sense that your body was betraying you in ways you couldn't understand or control. That psychological toll extended far beyond the physical loss. Women blamed themselves even though nothing they did caused the problem. Families fractured under the stress. Marriages ended. Some women simply stopped trying after 2 or 3 losses. Better to have one living child than keep risking pregnancies that end in heartbreak. That decision, repeated across thousands of families, would have created exactly the kind of selection pressure that should have eliminated Rs-negative blood from populations. Women who were Asi negative and married to Rh positive men would on average have fewer surviving children than Rh positive women in the same situation. Over many generations, that difference in reproductive success should have driven Rh negative frequencies down. The fact that it didn't, Rh negative frequencies remained stable or even increased in some populations. Either the reproductive disadvantage was smaller than it seems, or it was offset by advantages in other areas. Maybe Rh negative women had slightly better survival in childhood, balancing out later reproductive losses. Maybe Rh negative men had some advantage in attracting mates or surviving to reproductive age. Maybe in small communities with limited partner choice, blood type matching was less important than other factors in determining family size. Or maybe the story is more complex, involving multiple factors that varied across different populations and time periods. The Basques might have maintained high Rh negative frequencies for different reasons than the Irish or the Berbers. Population genetics rarely offers single simple explanations. Usually the answer is it's complicated with multiple forces pushing in different directions. The final result determined by whichever forces happen to be stronger in particular times and places. What we're left with is a trait that clearly matters medically, that clusters geographically in ways that suggest ancient population structure, that creates real challenges for reproduction, but has nonetheless persisted through countless generations. A trait that draws speculation and theory precisely because the straightforward explanations don't quite fit all the evidence. Something more is going on, something we haven't fully grasped yet. This uncertainty creates space for alternative theories, explanations that venture beyond mainstream science into speculation, and sometimes fantasy. When conventional answers leave obvious questions dangling, human imagination fills the gaps. This is how myths are born, how mysteries accumulate layers of interpretation that drift further and further from the original biological fact. The ancient alien theory represents perhaps the furthest reach of Rao's negative speculation. In this narrative, extraterrestrial beings visited Earth in prehistoric times and interbred with humans. The offspring of these unions carried genetic markers, distinguishing them from purely terrestrial humans. Rate 1 till N negative blood, according to this theory, is one of those markers, a flag identifying individuals who carry some fraction of alien DNA in their genome. Proponents point to ancient texts describing gods descending from the sky and mating with human women. They reference the Nephilim mentioned in Genesis, those mysterious beings born when the sons of God came unto the daughters of men. They ask why Rh-negative blood causes pregnancy complications, suggesting this incompatibility indicates mixture between two species, not meant to interbreed. They note the geographic clustering, proposing that ancient landing sites or settlement areas corresponded to regions where high Rh-negative frequencies persist today. The theory gains traction in certain circles because it offers a grand narrative, a story that makes Rh-negative individuals feel special, connected to something cosmic. Instead of being a random genetic variant, Rh-negative becomes a badge of hybrid ancestry, a marker of bloodlines touched by beings from beyond Earth. The pregnancy complications transform from medical problem to evidence of fundamental incompatibility between terrestrial. We can trace Rh-negative variants through human populations using standard population genetics. We can see where and approximately when the deletion happened. We can identify the specific genetic changes involved, deletions, disruptions, and mutations in the RHD gene that follow patterns consistent with normal evolutionary processes. There's no mysterious genetic material that doesn't fit with terrestrial origins. No sequences that look like they came from outside Earth's biological heritage. The pregnancy complications don't suggest species incompatibility. They reflect normal immunological processes. The same mechanisms that protect us from disease can sometimes create problems in context evolution didn't specifically optimize for. Pregnancy is challenging immunologically even in the absence of Rh incompatibility. The body must tolerate a genetically distinct individual, and this tolerance is achieved through carefully regulated immune suppression. When that regulation breaks down, problems occur. This happens in various contexts, not just Rh incompatibility. As for ancient texts describing sky gods and divine offspring, these can be understood as mythology, metaphor, attempts to explain natural phenomena before scientific frameworks existed. Every culture has stories of interactions between human and divine realms. These stories serve psychological, social, and spiritual functions. They don't constitute evidence of literal extraterrestrial contact any more than stories about dragons prove that fire-breathing reptiles once roamed the Earth. The Atlantis theory sits somewhere between ancient astronauts and mainstream science in terms of plausibility. Plato described Atlantis as an advanced civilization destroyed in a catastrophe around 11,000 years ago. If Atlantis existed, a big if its survivors would have dispersed to other lands, carrying their genetic heritage with them. Perhaps Rh-negative blood was common in Atlantis, and its high frequency in modern Basques, Irish, and Berbers reflects settlement. This theory at least proposes a terrestrial origin for Rh-negative, making it more compatible with known genetics than alien hypotheses. The problem is lack of evidence for Atlantis itself. Despite extensive searching, no one has found archaeological evidence of the advanced civilization Plato described. Most historians consider Atlantis either pure fiction or a garbled memory of historical events like the Minoan eruption at Thera, fictionalized and expanded for philosophical purposes in Plato's dialogues. If Atlantis didn't exist, then any theory depending on Atlantean refugees falls apart. You can't explain modern genetic distributions based on migrations from a place that never existed. And even if Atlantis were real, you'd need evidence that its population had unusual Rh-negative frequencies before you could claim it as the source of modern distributions. Speculation builds on speculation without foundation in verifiable evidence. The biblical interpretation focuses on the Nephilim, those beings mentioned briefly in Genesis. The text says they were mighty men, men of renown, born when divine beings mated with human women. Later passages mention giants, suggesting the Nephilim were unusually large. Some interpreters connect these passages to fallen angels, divine beings who rebelled against God and corrupted humanity before the flood. In this framework, Rus-negative blood becomes a genetic signature of Nephilim heritage. Perhaps the original Nephilim were Rus-negative, and that trait persisted in their descendants even after the flood killed most carriers. Perhaps Noah's family included Rus-negative individuals who carried the trait forward. Perhaps royal bloodlines preserved Rhi-negative frequencies through careful breeding, knowing they carried something special even if they didn't understand the genetic. This interpretation offers spiritual meaning to what might otherwise seem a random genetic variant. It connects modern individuals to ancient narratives, to stories of divine intervention and cosmic struggle. For people who take biblical accounts literally, it provides framework for understanding why this blood type exists and why it matters. But biblical texts don't provide genetic information. The Nephilim account is brief and cryptic, open to multiple interpretations. The word Nephilim might refer to giants, to fallen ones, to mighty men. Translations and interpretations vary. There's no mention of blood types or genetic markers. Drawing a line from Genesis to modern Rhi-negative distributions requires reading into the text details that simply aren't there. Moreover, if we're taking the flood account literally, then all modern humans descend from Noah's family, a genetic bottleneck far more severe than anything genetic evidence actually shows. Real population genetics demonstrates continuous human presence and genetic diversity throughout the period when the flood supposedly occurred. There's no evidence of humanity being reduced to a single family 10 to 15,000 years ago. These alternative theories persist not because evidence supports them but because they offer something mainstream science doesn't: narrative meaning, a sense of specialness, connection to grand cosmic or spiritual stories. They turn a medical fact into a badge of identity, a marker distinguishing carriers from ordinary people. They make the isolation and challenges some Rh-negative individuals report feeling seem meaningful rather than random. This psychological dimension matters. People want to understand themselves, to feel their experiences are valid and significant. When you discover you're part of a small minority, when you learn your blood can do things other people's blood can't, attack your own babies, universally donate in emergencies, it's natural to search for meaning beyond clinical descriptions. The alternative theories provide that meaning even if they don't provide valid explanation. The challenge is maintaining balance. You can acknowledge that Rho-negative blood is genuinely unusual, that its distribution raises interesting questions, that science doesn't yet have complete answers, without leaping to conclusions that evidence doesn't support. You can hold space for mystery, while still respecting the boundaries between speculation and established fact. Mainstream science's current best answer remains the most parsimonious. Rho-negative arose through mutation in European or Middle Eastern populations tens of thousands of years ago. It spread through normal population movements and increased in frequency in some isolated groups through genetic drift, possibly aided by modest selective advantages we haven't fully identified. It persisted despite pregnancy complications, because those complications weren't severe enough to eliminate the trait entirely, especially in populations with access to cultural knowledge about managing difficult pregnancies. This explanation doesn't invoke anything beyond normal evolutionary processes. It doesn't require aliens, or lost civilizations, or supernatural intervention. It simply acknowledges that sometimes evolution produces patterns that look strange from our limited temporal perspective, make sense within the full context of human population history spanning hundreds of thousands of years across multiple continents and dramatically changing environments. Yet, even this mainstream explanation leaves room for discovery. Future research might reveal subtle advantages that clearly explain why natural selection tolerated Rhi-negative despite its reproductive costs. We might find that it provided significant protection against specific pathogens that were major killers in ancient Europe, but have since been eliminated or become less virulent. We might discover metabolic or immunological benefits that gave Rhi-negative individuals slight edges in survival under Ice Age conditions. Or we might conclude that advantage played minimal role, that this really was mostly genetic drift amplified by small population sizes and geographic isolation. Not every trait exists for a reason. Some persist simply because they weren't harmful enough for selection to efficiently remove them. Evolution optimizes locally and gradually, not globally and instantly. Suboptimal traits can linger for thousands of generations if they don't cause immediate catastrophic problems. The pregnancy paradox was serious, but not immediately catastrophic in evolutionary terms. Women still managed to have surviving children even if they lost some to hemolytic disease. In societies where large families were normal, losing 2 or 3 children while raising 4 or 5 still allowed population maintenance. Tragic for the families involved, but not severe enough to drive rapid frequency changes through selection, and in small isolated populations, individual survival and reproduction depends on many factors beyond genetics. Was food available? Did disease strike? Did raiders attack? Did the winter prove especially harsh? Random environmental variation can overwhelm subtle genetic effects, making it difficult for selection to consistently favor one variant over another. Under those conditions, random sampling effects in which genes get passed forward becomes the dominant force shaping frequency changes. So perhaps Rh negative blood's greatest mystery isn't why it exists but why we find it so mysterious. Maybe the lesson is that not everything in biology has an obvious adaptive explanation. Some things just are, preserved by the stochastic processes that always operate alongside selection, maintained in gene pools through simple accident of inheritance in lineages that happen to survive. Yet that conclusion, while scientifically sound, satisfies neither the researchers who hope to find clear selection signatures, nor the individuals who experience being Rh negative as meaningful identity. Everyone wants stories to resolve cleanly, for questions to have definite answers. Science doesn't always cooperate with that desire. Sometimes the honest answer is we're not sure yet, or even we may never know for certain. What we do know beyond doubt is that Rh negative blood matters medically. It shapes transfusion compatibility, pregnancy management, organ transplant considerations. It influences how doctors approach prenatal care and emergency medicine. It affects research into blood substitutes and synthetic blood products. These practical implications ground the mystery in concrete reality, reminding us that whatever fascinating questions the trait raises about human history, it first and foremost affects real people's health and survival. The blood bank refrigerator with its carefully tracked Rh negative inventory represents this practical side. Every unit has an expiration date, a limited window of viability. Red blood cells can be stored cold for about 42 days before they degrade too much for transfusion. Hospitals must constantly balance their stock, using older units before they expire while maintaining sufficient reserves for emergencies. For Rh negative blood, this balancing act is more delicate, because replacement depends on a smaller donor pool. During mass casualty events, accidents, natural disasters, acts of violence, the O negative stock depletes fastest. Emergency rooms default to O negative when there's no time for typing and cross-matching, when seconds mean the difference between life and death. This creates surges of demand that blood banks must anticipate and plan for, even though predicting when disasters will strike is impossible. They maintain buffer stocks, call donors proactively, coordinate with other hospitals and regional centers to ensure supply matches need. Rh negative donors, especially O negative, receive calls regularly. Blood banks track them, know their donation history, reach out when stocks run low. Some donors give every 8 weeks like clockwork, treating it as civic responsibility. Others donate sporadically, responding to urgent calls but not maintaining regular schedules. The system depends on both types, on a network of individuals who understand their blood has special value and choose to share it. There's something profound in that choice. Donating blood is fundamentally about connection, about extending biological self to help strangers you'll likely never meet. The blood taken from your arm might flow into someone else's body during surgery, trauma, childbirth, cancer treatment. Your plasma proteins might help a burn victim maintain blood pressure. Your platelets might allow a leukemia patient to survive chemotherapy. The connection is intimate even though it happens through medical intermediaries and technical procedures. For Rh negative donors, this connection carries additional weight. Your blood fits fewer people, making each donation statistically more likely to serve someone for whom compatible blood is scarce. This doesn't make Rh negative donations more valuable in absolute terms. Every blood type is desperately needed, but it does create a particular kind of responsibility. If you have rare blood and can donate, your contribution addresses a specific vulnerability in the supply chain. Some donors describe feeling this responsibility deeply. They schedule donations around it, plan their lives to remain eligible, avoiding certain medications, maintaining healthy iron levels, staying well hydrated. Others view it more casually, donating when convenient, but not organizing their lives around it. Both approaches are valid. Donation is always voluntary, always a gift rather than obligation. But the sense of being part of life-saving infrastructure can be a powerful motivator for those who feel it. Meanwhile, researchers continue probing the boundaries of what we know about rat biology. Some work focuses on the protein itself. What does it do? Why is it on red blood cells? Every protein exists for reasons, performs functions, even if we don't understand them. The D antigen isn't just a label or marker. It does something in the body, plays some role in cell function or signaling or structure. Current evidence suggests Rh proteins may be involved in transporting ammonia or carbon dioxide across cell membranes, helping red blood cells manage the gases they carry. They might affect cell shape or flexibility, influencing how easily red blood cells squeeze through tiny capillaries. They might interact with other membrane proteins in ways that influence cell survival or immune recognition. The research remains preliminary, the functions not fully mapped. If we understood exactly what the D antigen does, we might better understand why its absence persists despite apparent disadvantages. Perhaps lack of the protein provides subtle benefits in gas exchange or cell flexibility. Perhaps it alters immune responses. Perhaps it influences something unexpected: brain chemistry, hormone signaling, metabolic efficiency through pathways we haven't yet traced. Every answer generates new questions. If right proteins transport gases, does their absence affect oxygen delivery in subtle ways? If they influence cell flexibility, does that matter for blood flow through small vessels? If they interact with immune signaling, could that explain reported differences in disease susceptibility? Each thread leads deeper into cellular biochemistry, into mechanisms operating at scales where direct observation is difficult and inference relies on indirect evidence from multiple sources. Other research examines health correlations, looking for patterns that might reveal hidden advantages or disadvantages of Rh homeostasis. This work typically uses large medical databases, searching for statistical associations between Rh type and various conditions. Cancer rates, cardiovascular disease, autoimmune conditions, infectious disease severity all have been examined for Rh-related patterns. The results are mixed and often contradictory. One study finds an association, another fails to replicate it. Small sample sizes, confounding variables, and publication bias complicate interpretation. Positive findings get published while negative results often don't, creating literature that may overrepresent significant associations while underrepresenting null findings. Meta-analyses try to correct for this by combining multiple studies, but they can only work with data that exists in published form. What emerges from this literature is that if ROMP status influences disease risk, the effects are generally small and inconsistent across populations. Much larger than blood type are factors like age, smoking, obesity, exercise, diet, genetics at hundreds of other loci. Rh status might nudge risk slightly in one direction or another for specific conditions, but it's not a major determinant of health outcomes for most people in most circumstances. This makes sense evolutionarily. If Rh negative provided huge advantages or imposed massive costs beyond pregnancy, we'd see much stronger frequency patterns. Traits with large effects on survival don't reach 30% frequency through drift alone. They're either strongly selected for or against. The fact that Rh negative frequencies vary widely but don't approach fixation or loss in most populations suggests the fitness effects are modest, easily swamped by other factors. COVID-19 research followed this pattern. Initial excitement over apparent blood type associations gave way to more muted conclusions as better data accumulated. Yes, there might be small effects. No, blood type alone doesn't determine your COVID risk in any meaningful practical sense. Get vaccinated, wear masks in high-risk settings, manage underlying health conditions. These matter far more than whether you're Rh positive or negative. Yet the question keeps drawing attention because identifying even small genetic risk factors can illuminate biological mechanisms. If type O blood provides modest protection against COVID, understanding why might reveal something about how the virus infects cells or how the immune system responds. Even if the effect is too small to guide individual behavior, it might point toward therapeutic targets or help explain why some people experience severe disease while others remain asymptomatic. This is true of many complex diseases. Heart disease risk varies with blood type, probably because ABO antigens affect blood clotting and inflammation in subtle ways. Understanding these mechanisms might eventually lead to better treatments, even though blood type alone doesn't determine who gets heart disease. Genetics rarely provides simple answers. This gene causes this disease, but it offers threads that, when followed, reveal underlying biology worth understanding. For Rh-negative individuals, following this research, the emotional experience can be frustrating. Every few months a headline proclaims Rh-negative blood linked to X based on a single study with modest sample size. Then follow-up studies fail to replicate the finding or show it applies only in specific populations. You're left unsure what to believe, whether your blood type really affects your health or whether these are statistical artifacts being amplified by media hungry for attention-grabbing stories. The reality is probably both. Some associations are real but small, others are spurious, products of chance variation in data sets that will disappear with larger samples. Distinguishing which is which requires patience, replication, careful analysis of multiple studies using different populations and methods. Science moves slowly towards certainty, testing and retesting until consensus emerges. Individual studies are rarely definitive. Truth emerges from the aggregate pattern. For people trying to make health decisions, this creates legitimate uncertainty. Should you worry about conditions that show weak associations with Rh-negative status? Should you mention your blood type when discussing health risks with doctors? The practical answer is usually no. Focus on factors with stronger evidence and larger effects. But the uncertainty remains because science hasn't yet provided clear definitive answers about what, if anything, Rh-negative means for health beyond pregnancy and transfusion. This is where alternative health communities sometimes step in, offering certainty that mainstream medicine can't provide. Various dietary recommendations, supplement regimens, lifestyle modifications get promoted specifically for Rh-negative individuals. These recommendations usually lack strong evidence but appeal to people seeking guidance, wanting to optimize their health based on their specific blood type. The blood type diet became popular through this mechanism, proposing that people should eat according to their blood type with different recommendations for OAB and AB. Serum status wasn't the primary focus, but some promoters extended the logic to include it. Eat this, avoid that, follow these principles, and you'll optimize health for your blood type. The concept sounds plausible. Biology does vary between individuals, so why not dietary needs? But when researchers tested blood type diet claims rigorously, they found no evidence supporting them. People don't actually experience better health outcomes eating according to their blood type compared to following generally healthy dietary patterns. The specifics proposed by blood type diet advocates don't correspond to real nutritional differences that vary by blood type. It's pseudoscience wrapped in biological language, appealing because it offers personalized recommendations but unsupported by actual evidence. This pattern repeats across various health domains. Claims proliferate faster than research can test them. Some people report benefits from following blood type-specific recommendations, but placebo effects and regression to the mean probably explain most positive experiences. When you start paying attention to your health, trying new things, you often feel better, regardless of whether the specific intervention actually works. Separating real effects from psychological ones requires controlled studies, not anecdotes. So we're left with uncertainty that's uncomfortable but honest. Rh-negative blood definitely matters for pregnancy and transfusion. It might have small effects on disease risk that research hasn't yet clearly characterized. It doesn't justify radically different health approaches or dietary patterns. The best advice remains standard: eat well, exercise regularly, manage stress, get preventive care, treat your body with basic respect. Your blood type doesn't override these fundamentals, yet the hunger for clear answers persists, driving both legitimate research and speculative theorizing. This tension between what we know and what we wish we knew creates the psychological space where Rh-negative blood becomes more than medical fact. It becomes identity, mystery, focal point for questions about who we are and where we came from. The science and the speculation circle each other, sometimes touching, often diverging, both trying to make sense of the same biological reality from different angles with different tools. The laboratory lights stay on through the night in research facilities, where scientists work with blood samples frozen in liquid nitrogen, preserved at temperatures so cold that biological time essentially stops. These samples come from people scattered across continents, volunteers who agreed to contribute their genetic material to studies trying to map human diversity. Among these thousands of samples, the Rh-negative ones get special attention, tagged in databases, analyzed repeatedly as new techniques become available. Researcher pulls up a screen showing genetic sequences from 50 Rh-negative individuals, spanning from Ireland to Iran. The screen fills with letters representing DNA bases, long strings of A's and T's and C's and G's. Somewhere in those sequences lies the answer to questions about ancient migrations, about when and where and how many times the RHD deletion occurred. The data is there, frozen in molecules, waiting for the right analysis to reveal its secrets. Modern genomic tools can do things that would have seemed like magic to the scientists who first discovered the Rh factor. They can sequence entire genomes in days. They can compare thousands of individuals simultaneously, identifying tiny variations that distinguish populations. They can estimate when mutations occurred by analyzing the patterns of variation surrounding them. They can trace ancestry backward through generations, watching genetic markers split and merge as populations diverged and reunited. When researchers apply these tools to Rh-negative blood, they find something interesting. The RHD deletion isn't a single event that happened once. Multiple deletions occurred independently in different populations, though the most common European variant does appear to stem from a single ancestral deletion. This deletion then spread through European populations, carried by people who moved and mixed and sometimes stayed isolated in mountain valleys where it reached unusually high frequencies. But there are other Rh-negative variants with different molecular signatures. Some involve different deletion patterns. Others result from mutations that disrupt the gene without fully deleting it. These variants are rare, usually specific to particular populations. They tell stories of independent genetic accidents happening in different places at different times, most remaining uncommon but a few spreading widely enough to leave detectable traces in modern populations. This complexity matters because it suggests Rh-negative wasn't a single evolutionary experiment, but multiple ones. If the same trait arose independently several times and persisted in multiple populations, that might hint at some advantage, some reason natural selection tolerated or even favored these variants despite the pregnancy complications. Random mutations happen constantly but most disappear quickly. For Rh-negative to arise multiple times and persist widely enough that we can detect it today suggests something more than pure chance. Yet the geographic pattern still points toward Europe as the primary source of high Rh-negative frequencies. The ancient DNA evidence confirms this. Samples from European hunter-gatherers show Rh-negative present but not dominant. Samples from early farmers migrating into Europe from the Middle East show mostly Rh. In Western Europe, where hunter-gatherer ancestry remained stronger, Rh-negative frequencies stayed higher. In Eastern and Southern Europe, where farmer ancestry dominated, frequencies were lower. This mixing created the gradient we see today, a steady decline in Rh-negative frequency. As you move from the Atlantic coast toward Asia, the gradient isn't perfectly smooth, interrupted by mountain ranges and language barriers and historical population movements, but the overall pattern is clear. Rh-negative clusters were ancient European hunter-gatherer ancestry clusters, suggesting the trait was more common in those populations than in the agricultural peoples, who eventually absorbed or replaced them across most of Europe. The Basque anomaly fits this pattern. Genetic studies show the Basque people carry unusually high amounts of ancient European hunter-gatherer ancestry. They're not pure descendants of pre-agricultural Europeans, nobody is, but they maintain a genetic profile suggesting less mixing with incoming agricultural populations than occurred elsewhere. Their language, completely isolated from surrounding Indo-European tongues, supports this interpretation. They represent a population that remained relatively isolated, preserving both linguistic and genetic patterns that vanished elsewhere. Whether this isolation was geographic, cultural, or some combination remains debated. The Pyrenees certainly create physical barriers, but valleys and passes exist. People could and did move through these mountains throughout prehistory and history. Yet somehow the Basque people maintained distinctiveness. Perhaps their early adoption of certain cultural practices, their social organization, or simply the ruggedness of the terrain they inhabited, allowed them to resist absorption longer than lowland populations. Whatever the mechanism, the result is visible in their genes and their blood. 35% Rh negative represents not a recent development, but ancient persistence, a window into what European blood type frequencies might have looked like before waves of migration from the east and south reshaped the genetic landscape. The Basques aren't unique in evolutionary terms, just unusually conservative, holding on to patterns their neighbors lost through mixing. This interpretation transforms how we think about Rh negative blood. It's not alien or divine or specially evolved for some hidden purpose. It's simply old, a marker of ancient European ancestry that survived in some populations and faded in others as human groups mixed and migrated over thousands of years. The mystery isn't why it exists, but why it stayed common in specific places while becoming rare elsewhere. Part of that answer certainly involves geography. Mountains, islands, peninsulas, regions with limited access naturally slow genetic mixing. The Scottish Highlands maintained high Rh negative frequencies for the same reasons they maintained Gaelic language and clan structures longer than surrounding areas. Ireland's island status created similar isolation. The Atlantic fringe of Europe, from Portugal to Norway, represents a zone where older genetic patterns persisted because fewer people migrated through, compared to Central European plains crossed repeatedly by armies, traders, and settlers. But geography alone can't fully explain the pattern. Iceland is an island, more isolated than Ireland, yet its Rh negative frequency is moderate rather than extreme. The Americas remained isolated from Europe for thousands of years, yet indigenous populations there show almost no Rh negative. Geographic isolation preserves existing variation but doesn't create it. For Rh negative to reach high frequencies requires either that it was already common when populations became isolated, or that it increased through drift or selection after isolation occurred. The timing suggests both factors played roles in different places. Among the Basques, Rh negative was probably already fairly common in ancestral populations, then maintained through relative isolation. In some Celtic populations, frequencies might have increased through drift in small founding groups that colonized islands or mountain regions. In other cases, local selection pressures we don't fully understand might have favored Rh negative individuals for reasons specific to particular environments or disease ecologies. This patchwork explanation lacks the satisfying simplicity of a single grand theory, but it probably comes closest to truth. Human population history is messy, shaped by countless individual events and local circumstances that played out differently in different places. Genes like the RHD deletion get carried along by the people who bear them, rising and falling in frequency according to population size, migration patterns, isolation, and occasionally selection, but mostly through the random sampling that occurs when populations expand and contract and divide and merge. If you could watch European genetic frequencies change over the past 40,000 years like a time-lapse video, you'd see waves of variation spreading and contracting. Rh negative appearing in hunter-gatherer populations, spreading somewhat, then getting diluted by incoming farmers, then persisting at high levels in pockets where mixing was limited, then shifting again as medieval and modern migrations reshuffled European genetics yet again. The current distribution represents a snapshot of a process still ongoing, frozen arbitrarily at the present moment, but continuing to evolve as populations continue mixing. In another 1,000 years, Rh negative frequencies will likely have shifted further. Increased mobility and intermarriage between populations that were historically isolated will tend to homogenize frequencies toward global averages. The Basque anomaly will probably diminish as younger Basques increasingly marry non-Basques and their children carry different genetic combinations. This isn't loss of diversity exactly, just redistribution, the same genes spreading more evenly rather than clustering intensely in specific regions. Yet certain factors could work against homogenization. The Rh-negative individuals preferentially partner with other Rh-negative individuals either through conscious choice or because shared ancestry creates social networks where marries then high-frequency pockets could persist longer than random mating would predict. If cultural revival movements strengthen Basque or Celtic identity and create preferences for within-group marriage, genetic distinctiveness might be maintained deliberately rather than eroding through default mixing. And if it turns out that Rh-negative does provide genuine health advantages or disadvantages that modern environments make newly relevant, selection could start shifting frequencies in ways we can't currently predict. Perhaps some future pathogen will emerge against which Ra negative individuals show better resistance, driving frequencies upward. Perhaps some environmental change will make the pregnancy complications more severe, increasing selection against the trait. Evolution hasn't stopped just because we have modern medicine. It continues operating, though often in ways too subtle or too slow for us to directly observe. For the individuals carrying Rh-negative blood today, these deep-time perspectives can feel both grounding and dizzying. Your blood type connects you to specific ancestral populations, traces lineages reaching back tens of thousands of years to people who lived in vastly different worlds. Stand on a hillside in the Basque Country or the Scottish Highlands, and you're walking where ancestors with your same blood type walked, hunting and gathering and eventually farming the same landscapes, dealing with cold and hunger and danger using tools and knowledge systems we can barely reconstruct. That connection is real, written in molecules that get copied and passed down through generations with remarkable fidelity. The same genetic instructions that shaped red blood cells in someone's body 30,000 years ago shaped yours, transmitted through an unbroken chain of parents and children, each generation passing the torch forward without break or interruption. If that chain had broken even once, if any one of your direct ancestors had failed to reproduce, you wouldn't exist and your particular genetic combination would never have formed. This is true for everyone regardless of blood type. But Rh-negative creates a specific kind of connection because its rarity and geographic clustering. If you're Rh-negative and your ancestry traces to Western Europe, you're carrying a marker strongly associated with ancient European hunter-gatherers. Not exclusively, the trait exists in other populations and can be transmitted through any lineage, but statistically, probabilistically, your Rh-negative status hints at connections to populations that lived in Europe before agriculture arrived. This doesn't make you special in any meaningful sense, doesn't grant abilities or mark you as chosen. It's simply historical information encoded in your genome, one data point among millions that collectively describe your ancestry. But for people interested in genealogy and deep origins, blood type can provide clues about which migration paths their ancestors followed. Commercial ancestry testing companies have started incorporating blood type information into their analyses, not because blood type is hugely informative. It's a single locus while they analyze hundreds of thousands, but because customers find it interesting. The reports might say something like your Rh-negative status is most common in populations from Western Europe, consistent with significant ancestry from that region. Not definitive proof of anything, just consistent patterns that add texture to the larger ancestry picture. For adoptees or people whose family history was disrupted by war, migration, or social circumstances, even these small clues can feel meaningful. Learning your Rh-negative might be the first concrete biological connection you've identified to specific geographic regions or populations. It's a tiny thread, but sometimes tiny threads are all you have to start reconstructing lost histories. The medical reality of being Rh-negative remains the most concrete dimension of this experience. You carry a card in your wallet, or remember the designation. Know to tell doctors during medical encounters, understand that if you're female and might become pregnant, this status matters significantly. These practical implications ground the trait in present-day life rather than abstract genetics. Blood donation becomes a regular reminder of that status for those who choose to donate. You arrive at the donation center. They check your eligibility, stick your finger for a quick hemoglobin test, then settle you into a chair and find a vein. The needle slides in. Blood begins flowing into the collection bag, and you spend the next 10 minutes squeezing a ball occasionally while dark red liquid accumulates slowly in plastic. It's a strange feeling watching your blood leave your body, knowing it will go into someone else's veins, become part of their circulatory system, keep them alive through whatever crisis brought them to need it. The donation center staff treat Rh-negative donors with particular appreciation. They might mention that your type is especially needed, that emergency rooms depend on Rh-negative for trauma cases, that premature babies often need Rh-negative blood. This knowledge can make the mild discomfort and temporary fatigue feel worthwhile, transform a slightly unpleasant medical procedure into an act of connection with strangers you'll never meet, but whose lives might depend on what flows through your veins. Some donors describe it as their civic duty, the equivalent of voting or jury service, but biological rather than political. Others see it in more personal terms, giving back to a system they or family members might depend on someday, paying forward the gift of life that medicine makes possible. If you approach it spiritually, viewing blood donation as sacred act, literal sharing of life force with those in need, whatever the motivation, the aggregate effect is crucial infrastructure. The blood supply depends entirely on voluntary donors willing to give regularly enough that hospitals never run dry. For Rh-negative types, this dependency is more acute because the donor pool is smaller and the need just as urgent. A single car accident involving multiple Rh-negative patients could exhaust a regional blood bank's O-negative stock in hours. Maintaining adequate supplies requires constant recruitment and retention of donors willing to give repeatedly over years. Blood centers use various strategies to keep donors engaged. Regular reminders through text or email about when you're eligible to donate again. Thank you messages telling you what hospital received your blood. Milestone recognition when you reach certain donation totals. Small gifts, juice and cookies, and occasional prize drawings. Anything to keep people coming back, to transform one-time donors into regular contributors whose donations can be counted on in planning supply levels. For Rh-negative donors, the psychological reward of being particularly needed can itself motivate regular donation. Knowing your blood is scarce makes giving feel more significant, turns you from one anonymous donor among thousands into someone whose contribution addresses a specific gap. This isn't necessarily accurate. Every blood type is needed, and O-positive donors probably contribute more total volume simply because there are more of them, but the perception of special need can drive behavior, even when the underlying reality is more complex. The blood bank refrigerator represents the end result of this entire system. Row after row of plastic bags hanging from metal racks, each labeled with type and collection date, each representing one person's gift and another person's potential lifeline. The Rh-negative section stays carefully monitored. Older units rotated forward to be used before they expire. New units slotted into place as donors arrive. The whole inventory tracked in computer systems that alert staff when levels drop below safe thresholds. During normal times, this system works smoothly enough that most people never think about it. Blood is available when needed, surgeries proceed on schedule, trauma patients receive transfusions quickly. The infrastructure runs invisibly in the background, one of countless systems making modern life possible without calling attention to itself. But during crises, the fragility becomes apparent. Major disasters can exhaust blood supplies within hours as casualties flood emergency rooms. Severe weather that prevents donors from reaching collection centers can create shortages. Disease outbreaks that make people reluctant to visit donation centers or that increase demand from hospitalizations can strain supplies. The COVID pandemic created both problems simultaneously: fewer people willing to donate during lockdowns combined with high demand from severely ill patients. During these crises, blood banks issue urgent appeals through media and social networks. They particularly emphasize need for O-negative and other Rh-negative types, knowing those supplies deplete fastest during mass casualty events. The appeals usually work. Disasters motivate people to help, but the surge of donations takes days to materialize while the need is immediate. Maintaining buffer stock becomes crucial but also expensive, since blood expires relatively quickly. This ongoing tension between supply and demand, between perishability and need for reserves, drives research into blood alternatives and synthetic blood products. If we could manufacture blood in laboratories or develop substitutes that carry oxygen without requiring human donors, we could eliminate the supply problems that occasionally create life-threatening shortages. Blood would become a manufactured product rather than a gift, always available in whatever quantities needed. The technology isn't there yet. Lab-grown red blood cells exist in research settings but remain far too expensive and difficult to produce at the scales needed for clinical use. Oxygen-carrying substitutes have been developed but tend to have side effects that limit their use to specific emergency situations. Whole synthetic blood that replicates all the functions of natural blood remains beyond current capabilities. So for now and for the foreseeable future, the blood supply depends on human donors. Those with Rh-negative blood carry their particular role in this system, their particular contribution to keeping the infrastructure functional. It's easy to forget this connection in daily life, to think of your blood as simply the liquid keeping you alive rather than as a potentially life-saving resource for others. The blood bank card in your wallet serves as a reminder of that dual nature. This blood is yours, but it's also something that could belong to someone else if circumstance and generosity bring it to that. The scientific questions continue in parallel with these practical realities. Researchers probe deeper into what the RHD protein actually does, using cell cultures and animal models to test hypotheses about its function. The current understanding suggests it may be involved in moving carbon dioxide or ammonia across red blood cell membranes, helping cells manage waste products from metabolism. If true, this would mean Rh-negative individuals handle gas transport slightly differently than Rh-transportive individuals, potentially with subtle effects on efficiency or performance, but the evidence remains incomplete. The protein sits in the membrane alongside dozens of others, all interacting in complex ways. Teasing apart the specific contribution of RHD from the contributions of its neighbors requires careful experimentation. Knockout experiments where scientists eliminate the gene in cell lines or organisms provide clues, but cells often compensate for missing proteins by upregulating alternatives, making it hard to isolate the effect of any single component. Human studies comparing Rh-positive and Rh-negative individuals on various physiological measures might reveal functional differences if they exist. Do Rh-negative individuals show different blood gas levels during exercise? Different altitude tolerance, different metabolic efficiency? These questions require large studies with careful controls because individual variation is enormous. So far, such studies haven't revealed dramatic differences. If Rh-negative status affects gas transport or metabolism, the effects are small enough to be hidden by normal variation between individuals. This doesn't mean there's no effect, just that detecting it requires either larger sample sizes or more sensitive measurements than have been employed so far. Other research examines whether Rh proteins interact with immune signaling pathways. The immune system uses the cell surface as a communication network, with proteins binding and unbinding and changing shape to transmit information about threats and responses. If RHD participates in these signaling networks, its absence might alter immune function in ways that could explain reported differences in disease susceptibility. Again, evidence remains preliminary. Some studies suggest associations between Rh type and immune-related conditions, but results are inconsistent across populations. The immune system is fantastically complex, involving thousands of genes and constant environmental influences. Detecting the contribution of any single protein requires studies large enough and detailed enough to control for countless confounding factors. The uncertainty frustrates both researchers and the public. We live in an era of rapid scientific progress where new discoveries happen constantly, where genome sequencing became routine and CRISPR gene editing moved from science fiction to laboratory reality within a few decades. Against that background, the fact that basic questions about Rh-negative blood remain unanswered. We've mapped the human genome, we've landed robots on Mars. Surely we can figure out what one missing protein does and why it persists. But biology resists simple answers. Living systems are irreducibly complex in ways that make prediction and explanation difficult, even with powerful tools. You can sequence every gene, but still not understand how they interact to produce organisms that develop and function and reproduce. You can identify every protein but still not grasp how they coordinate to create cells that somehow know when to divide and when to die and when to differentiate into specialized types. The RHD question sits within this larger biological complexity. Yes, we know the gene sequence. Yes, we know the protein structure. Yes, we know roughly where it sits in the cell membrane. But we don't fully know what it does, how its absence affects physiology, why evolution has tolerated or favored that absence in specific populations. Those deeper questions require not just sequencing and microscopy but functional studies, population studies, evolutionary analyses, all of which are difficult and expensive and time-consuming. Science progresses, but not always in straight lines or at uniform speed. Sometimes decades pass between identifying a phenomenon. Sometimes explanations remain provisional, the best current understanding rather than definitive truth. The Rh-negative story illustrates this perfectly, discovered in the 1930s, medically managed by the 1960s, but still not fully understood nearly a century later despite enormous research effort. Of individuals carrying Rh-negative blood. This scientific uncertainty creates psychological space where personal meaning can accumulate. When experts can't provide complete answers, people generate their own explanations. mixing fragments of science with intuition and experience and cultural narratives. The result is the rich folklore that surrounds Rho-negative blood, stories about special abilities and ancient origins that science neither fully confirms nor completely rules out. The tension between scientific uncertainty and personal experience becomes particularly acute around reported psychological and physical differences. Many race-negative individuals insist they feel different, experience the world differently, have sensitivities and intuitions that others don't share. Are these real biological differences traceable to RhD status? Are they psychological effects of believing you're part of a special minority? Are they cultural patterns learned from online communities where Rh-negative identity gets reinforced? Probably all three operating simultaneously. Impossible to fully disentangle. Consider empathy, frequently mentioned in lists of Rh-negative traits. Do Rh-negative individuals actually show higher empathy in controlled studies? The research that exists is limited and shows mixed results. Some studies suggest possible correlations, others find no differences. But even if studies conclusively showed no average difference, that wouldn't invalidate the lived experience of Rh-negative individuals who feel they're unusually empathic. Individual variation is enormous, and any individual might be highly empathic for reasons having nothing to do with blood type but still experience that empathy as connected to their Rh-negative identity, because that's how they've learned to interpret it. The same logic applies to other reported traits: sensitivity to light, unusual eye colors, lower body temperature, enhanced intuition. Some might have biological basis related to genes that happen to cluster with RhD deletions in certain populations. Others might be psychological and cultural constructions, real experiences interpreted through the lens of blood type identity. Separating these possibilities requires research that mostly hasn't been done: longitudinal studies following large numbers of Rh-positive, measuring traits carefully before people learn their blood type and develop beliefs about what it means. Such studies would be expensive and difficult, but might finally answer whether there's substance to the trait lists circulated in online communities or whether they represent confirmation bias and self-fulfilling prophecies. Until those studies happen, we're left with anecdotes and personal testimonies that feel true to those who experience them but don't constitute scientific evidence in the usual sense. The challenge is honoring people's experiences while maintaining scientific rigor. When someone says, "I'm Rh-negative and I've always felt different," that feeling is real and meaningful to them. Dismissing it as imagination or delusion shows disrespect, but accepting it as proof that Rh-negative blood causes distinctive psychological traits requires evidence beyond individual testimony. Both realities can coexist. Your experience is valid, and the explanation you've adopted might not be accurate. This nuanced position satisfies neither skeptics who want to debunk all Rh-negative mysticism nor believers who want scientific validation of their experiences. It sits uncomfortably in the middle, acknowledging mystery, while refusing to fill it with explanations that exceed available evidence. But that middle ground might be the most honest place to stand, the position that best reflects both what we know and what we don't. What we know with certainty is that blood moves through your veins right now, carrying oxygen to every cell in your body, that this blood is type and characteristics determined by genes you inherited from parents who inherited them from their parents in an unbroken chain reaching back to the origin of your species. That whether your red blood cells carry the D antigen or lack it, they're performing the same essential function, keeping you alive with every heart. What we know is that if you're Rh-negative, your blood creates specific medical considerations during pregnancy and transfusion, that modern medicine has solutions to these problems, that your blood type affects how doctors approach your care in certain situations. These realities are concrete and undeniable. What we know is that Rh-negative blood clusters geographically in patterns reflecting ancient population movement, that your blood type carries historical information about where your ancestors lived and how they moved across landscapes over thousands of years. This connection to the past is real even if its meaning is ambiguous. What we don't know is whether RhD status affects your personality, your abilities, your health in ways beyond the well-documented pregnancy and transfusion issues. We don't know if there are genuine psychological or physiological differences that science simply hasn't detected yet, or whether the reported differences reflect cultural and psychological factors rather than biology. We don't know with certainty why natural selection tolerated wrong negative blood, despite its reproductive costs, or what advantages, if any, offset those costs. These gaps in knowledge create the space where mystery lives, where speculation flourishes, where personal meaning can attach to biological fact. That space isn't going away soon. Even as research continues, even as our understanding deepens, some questions will likely remain difficult to answer definitively. Biology is messy, and humans are complicated, and the intersection of the two resists simple explanations. Perhaps that's okay. Perhaps not everything needs to be fully explained and catalogued and understood. Perhaps some mystery in our lives is valuable, reminding us that we don't have complete knowledge, that surprises and discoveries still await. The blood flowing through your veins has carried humans through hundreds of thousands of years of evolution, through ice ages and migrations and near extinctions and eventual global spread. It carries you now through this moment, this night, this quiet time of rest and reflection. Whether that blood is ROS positive or negative, common or rare, it's performing miracles with every cycle through your circulatory system. Oxygen binding in your lungs, releasing in distant tissues, carbon dioxide collected and returned for expulsion, nutrients distributed, wastes removed, immune cells circulating on patrol, clotting factors ready to seal breaches. All of this happening automatically, beneath consciousness, the gift of billions of years of evolution refining the systems that make complex life possible. Your blood type is one small detail in that vast biological inheritance. Important in specific contexts, fascinating for what it reveals about human history and population genetics, but ultimately just one note in the symphony of processes maintaining your existence. The Reich factor matters, but it doesn't define you. You're made of countless genetic variants, epigenetic modifications, environmental influences, experiences, choices, and chance events that collectively created the person listening to these words right now. So as you drift towards sleep, you can let go of questions about where Reich negative blood came from or what it means beyond the concrete medical facts. You can release speculation about ancient origins and special abilities. You can simply rest in the knowledge that your blood, whatever its type, is doing its quiet work, keeping you alive, connecting you to ancestors and descendants, in an unbroken biological chain that stretches across time. The heart beats without your conscious direction. The lungs breathe whether you pay attention or not. Blood circulates through vessels totaling thousands of miles in length, reaching every cell in your body, bringing what they need and removing what they don't. This happens in everyone, regardless of blood type, this fundamental process of being alive in a body that knows how to maintain itself. In hospitals right now, donors' blood is saving lives. The O-negative units in refrigerators wait for their moment of crucial need. RI-negative mothers carry healthy babies protected by ROGAM from the immune reactions that once killed so many. Researchers peer at blood cells under microscopes, trying to understand what proteins on their surfaces actually do. All of these activities continue in the night, quiet threads in the larger fabric of human life and medicine and science. Your role in this system is simply to exist, to carry forward the genetic heritage you received, to perhaps donate if you're able and willing, to appreciate the complexity of the biology keeping you alive. Whether Rh negative makes you special in some cosmic sense doesn't really matter. You're already special by virtue of being alive, by being the unique combination of genes and experiences that makes you specifically you. The questions will still be there tomorrow if you want to pursue them further. The research will continue, slowly filling in details of the picture. The mystery won't disappear completely. Some aspects of biology may always resist full explanation, but that's part of being human in a universe that doesn't always offer clear answers to our questions. For now, in this quiet moment, you can let it all settle. The molecules flowing through your cardiovascular system doing their ancient work, the proteins on cell surfaces marking you as self, the immune system maintaining surveillance, the oxygen feeding your brain as thought gives way to dream. All of it continuing without requiring your attention or control. The gift of being alive in a body that knows its work and performs it faithfully, beat after beat after beat. Rest easy in that knowledge. Your blood carries you through this night and into tomorrow, rise positive or negative, common or rare, marked by history but not bound by it. Just blood doing what blood has done for millions of years, keeping life alive, one heartbeat at a time. The story of wrong negative blood is ultimately the story of human survival, adaptation, and the beautiful complexity that emerges when evolution works with the materials at hand across deep time. It's written in molecules but experienced in lives, in pregnancies managed carefully, in donations given freely, in questions asked and sometimes answered and sometimes left open for future generations to solve. As sleep approaches, you can release all of it. Let the questions fade. Let the stories dissolve into the darkness. What remains is simpler breath moving in and out, heart maintaining its rhythm, blood flowing where it needs to flow. The fundamentals of being alive, shared by everyone regardless of blood type or ancestry or any other classification we might use to divide and categorize human diversity. In that fundamental sameness, that shared biological inheritance, perhaps we find the truest answer to questions about what makes us special. Not our blood type, not our genetic quirks, but our participation in the ongoing adventure of life on Earth, the 4-billion-year experiment that produced consciousness and curiosity and the capacity to wonder about our own origins. Your Rh-negative blood, if you carry it, makes you part of a specific story about human population history and migration and adaptation. But you're also part of a much larger story about carbon-based life, about DNA and evolution, about matter organizing itself into patterns capable of looking back at their own history and asking why and how and what it all means. That larger story continues as you sleep, as cells divide and proteins fold and chemical reactions proceed according to laws written into the fabric of the universe long before Earth existed. You're a temporary expression of those laws, a pattern that emerged and will eventually dissolve, but that exists now, tonight, in this moment of awareness, fading into rest. Let it all go. The RHD gene and its deletions, the pregnancy paradoxes and transfusion protocols, the geographic distributions and evolutionary pressures. Let it settle back into the quiet place where biology does its work without fanfare or explanation. Your blood knows what to do. It's been doing it your whole life and will continue until life ends, requiring nothing from you except the permission to keep flowing. Sleep comes like a tide, natural and inevitable. Blood continues its circuits, oxygen feeding dreams, waste products cleared away. The body maintains itself through the night, all systems coordinating in ways too complex to ever fully comprehend but simple enough that they happen automatically, reliably, magnificently. In the morning, questions might return. But for now, in this night that welcomes you toward rest, there's only the quiet fact of being alive, breathing, heart beating, blood flowing. Rh-negative or positive, ancient lineage or recent admixture. It all dissolves into the simple truth of existence, continuing, moment by moment, breath by breath, heartbeat by heartbeat. Good night.